Hello students. Now I'm going to provide the last set of videos covering plasmids which are extensively used in plants, followed by a short video about animals. Earlier to that we discussed about various vectors which are designed to carry out recombinant DNA technology in prokaryotes, followed by eukaryotes. So in case of prokaryotes, we discussed about the plasmids, then we also discussed about the cosmid vectors, and both in phage as well as in bacteria. Then we switched over to eukaryotic vector. So in case of a eukaryotic vector, we discussed about the major differences between prokaryotes and eukaryotes. In case of eukaryotes, the genetic material is in an organelle called nucleus covered by a nuclear membrane, unlike in a prokaryotes in fact. So then we have to have a totally different strategy. So then we chose yeast as a host organism, followed by plants and animals. So in case of a yeast, I think we discussed about what type of vectors are there. There are integration vector. I think you know that in case of an integration vector, so we need to have a homologous region in that the repetitive units are very critical. So then we followed the replication vector. We used the autonomous replication sequence of an yeast. Then the episomal vector, we took the advantage of 2 microplasmid and centromeric vector. So in order to during the cell division, so that the whatever the integration we have designed should be a stable one. And finally we discussed about the artificial chromosome. So now yeast is an eukaryote, and from the yeast we will see how the whole process can be extended to a plant. So in case of a plant, I think they use a terminology called as a plant breeding. You all know that in the world the population is increasing every day, and because of the urbanization, the agricultural land also decreases in fact. So the most challenging for any government is to supply food for this increased population. So several scientists, so by the process of plant breeding, so they have brought enormous success in the improvement of yield per acre. So everybody remembers, especially the botanist, an American scientist called Norman Borlaug. So he is the one, with the help of plant breeding, has developed several varieties. For example, his success is with the development of semi-dwarf, high-yielding, disease-resistant wheat. So he has coined the term Green Revolution. Because of this Green Revolution, I think most of the countries are sufficient with food. They don't have to depend on any other countries for food. So he is the one who developed this kind of a variety of wheat by the process of plant breeding, and he is also called as the father of Green Revolution. Whatever the variety he has developed, he has given to countries like Mexico, India, Pakistan. In case of Mexico, currently because of this variety, now they export wheat to most of the countries. India and Pakistan are also quite successful in our own food supply. In India, like Norman Borlaug, we also remember a famous scientist, M.S. Swaminathan. So he is also called as a father of Green Revolution in India. So he was very successful in the implementation of this wheat variety in India. Apart from that, he also developed varieties with respect to the rice. So now even his varieties of rice will provide good amount of yield per acre. So we all know about the success is because of this plant breeding. So now most of the breeding techniques are conventional. We also call this as a classical breeding. Now in case of a classical breeding, there are limitations. The major limitation is going to be that this is time consuming, and they also have to experience species barrier. The species barrier, again, if you want to combine a high yielding variety versus a disease resistant variety, if you want to combine these two species, then there is a barrier which is going to be a critical one. In fact, to overcome that, they have developed new technique. The new techniques, they are called as a somatic cell fusion. Another one is going to be the recombinant DNA technology. With respect to the recombinant DNA technology, we discussed about a TI plasmid. So we will just see some of the procedural aspects which is common in somatic cell fusion. Another one is going to be the recombinant DNA technology. So what exactly we are going to do in somatic cell fusion? So in somatic cell fusion, it is possible to mix group of genes or the chromosomes of different species. We can bring them together in a given cell. All right. Now from this particular one, so because of the fusion, the somatic cell fusion, now the somatic cell fusion can be, in the laboratory conditions, can be developed into a plantlets. We will just listen some of these terminologies in the coming discussion. In fact, another one is going to be the recombinant DNA technology. The difference between somatic cell fusion and recombinant DNA technology is the introduction of specific genes, not the entire group of genes or chromosomes. So in case of a somatic cell fusion, for example, there are two varieties. One is a high yielding variety and probably prone to diseases. Another one is a disease resistant variety. So we can mix them and develop a totally new variety. Such things are possible in somatic cell fusion. Whereas in recombinant DNA technology, example is going to be the famous one is the Bt cotton. A toxin gene has been introduced into the cotton variety so that it can be resistant from the pest. Simultaneously, we also have a Bt brinjal. Now we also have a golden rice. So in case of a golden rice, we all know that rice is principally a starch based material, and by the introduction of a group of couple of genes with respect to the production of vitamin A has been introduced. Now the golden rice along with starch do also contain vitamin A in it, in fact. So some of these things are possible in the plant breeding. So now we know, I think with an example, what exactly is a somatic cell fusion and what exactly is a recombinant DNA technology. We will study few examples how the whole process is done in case of a somatic cell fusion, and we also discuss about like how genes are introduced in the recombinant DNA technology. So in both case, somatic cell fusion as well as in the recombinant DNA technology, we need a host. For example, in prokaryotes, we discussed about gram-positive bacteria, gram-negative bacteria, then yeast as a host. Now here, the plant cell is going to be the host. And how are you make this plant cell amenable for the manipulations? So now how this plant cell can be acted as a host, in fact. So now that is the process of competence, in fact. Now how are you going to make the plant cell a competent one for all these manipulation? So we are going to prepare a plant cell into a protoplast. The protoplast is nothing but it's a plant cell lack of cell wall. So we do have the cell is covered by the cell membrane, and now the cell membrane is surrounded by the cell wall. And cell wall is highly impermeable for any manipulation, in fact. So we have to remove this cell wall. So now the removal of cell wall is by the enzymatic treatment. We all know that cell wall is made out of the cellulose. and we remove the cellulose by treating the enzyme cellulase. We will discuss couple of those technologies in the coming discussion. So a protoplast is nothing but a plant cell devoid of cell wall. So once we do have the protoplast, then using this particular protoplast or a plant cell without a cell wall, so with the help of polyethylene glycol in the presence of calcium chloride, DNA can be, or a group of the cell fusion is possible, in which the genetic material can be easily introduced. So now that is what is called as a protoplast. In fact, we will see how these protoplasts are useful in the somatic cell fusion as well as in recombinant DNA technology. So now, we will discuss about more about this protoplast and its application in the plant breeding. So now the protoplasts are extensively used in propagation, especially in the tissue culture. So we are going to use the terminology plant tissue culture as well as plant cell culture. So now in case of with a protoplast, we can also manipulate in such a way that a particular alkaloids of therapeutics can be increased by exposing this protoplast to various agents. So now we can increase the production of alkaloids or amino acid derivatives. So many things can be manipulated with the protoplast. Later, this protoplast with the can be grown in a controlled medium, and we can get into an individual plants. All right. Now, whatever the protoplast we discuss and whatever the manipulation we are talking right now, it is easy to do in dicotyledons, vegetables, nuts, fruits and flowers. So they got good number of success. So from these dicotyledon plants and from the protoplast, manipulations are much easy. But we do find several problems in the monocotyledons. So basically the agricultural product from the monocotyledons are going to be the wheat, rice, oats, barley. They have done some success, but it is easy to do the manipulations in the dicots rather than in a monocot. Now we know what is a protoplast. So from this particular protoplast, so now we will discuss our we can also we take the protoplast, do the manipulation. When you transfer this into a laboratory medium, a nutrient medium, so we are going to get a mass of cells instead of a plantlets. We are going to call that as a callus, also called as a callus culture. Now this is going to be done from any part of a plant. All right, now this is a callus culture. Now this one is going to be an undifferentiated mass of cells. All right, but in contrary, in case of a recombinant DNA technology, so we are also going to work with a neoplastic crown gall disease. So now we are going to develop an undifferentiated mass of cells from a plant. So now we call this as a callus. So with the help of a microorganism infection, so we are going to develop, not in a laboratory conditions, but in the natural conditions, a similar growth, a neoplastic growth of mass of cells. So now we term this as a crown gall disease. We will discuss like what exactly is a crown gall disease. So we will just discuss about the somatic cell fusion, and now I think we have come to the terminologies callus. Okay, so now we are going to extensively use this in a plant tissue culture. So now how are you going to develop this callus? So now we are going to grow this particular undifferentiated mass from, we are going to use a term called as an explant. So now it is just like a biopsy in case of an animal world. So now we are going to use these explants. What are these explants? Now explants are a region where the cell division is maximum. For example, root tip, buds, nodal segments, as well as in case of the seeds, in fact. So we are going to select several actively dividing region. Okay, take out a small piece. Now that one we call this as an explant. All right. So now we take this explant. So it is an actively dividing region of a plant. So we take this particular tissue and we are going to process like this. First thing is going to be that we take these explants. We know what are these explants. We wash thoroughly in distilled water. Then we subject for a disinfection. So what is known as a disinfectant. So usually the disinfectant are treated with sodium hypochlorite, hydrogen peroxide or mercury chloride. We can use any one of them so that for the disinfections. Later, so we are going to keep these washed disinfected explant on a nutrient medium. Again, in case of a plant nutrient medium, we have a solid medium as well as a liquid medium. So for a callus, better to have it in a nutrient, that is a solid medium. So we do have the basic nitrogen supply, carbon source and some minerals, salts, everything. But here we also have to use some plant hormones. So the commonly used plant hormones are cytokinin, auxin, gibberellins. So for the callus culture, we always make sure that the solid nutrient medium do contain low amount of cytokinin, auxin, gibberellin. Now what happens is that this particular explant, it is an undifferentiated actively dividing cell. So now this started multiplying. So now that will become a mass of cells. So because it doesn't look like a plantlet at all. So it doesn't look like a bud or doesn't look like a root. It is just an undifferentiated cell mass. So it started growing like a cell mass. So now that one is called as a callus, and we are going to prepare this callus on a solid nutrient medium with low concentration of plant hormones like cytokinin, auxin and gibberellins, in fact. So in the next we will discuss the schematic representation of various steps involved in a plant cell culture or a tissue culture. I told you that we have taken the explants, that is the shoots, roots, buds, seeds, whatever we discussed. Now we are transfer this particular explant on a nutrient solid medium. I think now under the low concentration of auxin and low concentration of cytokinin, so now this explant will started to grow, multiply and becomes a mass of cell, what we call this as a callus. All right. So if you do supply good amount of nutrients, I think the callus do grow. Now again, from this particular callus, so we don't do any more explant. You can take this callus, a part of the callus, when you inoculate them into a new medium, again it will grow into a callus. So in other words, a callus can be maintained, this particular stated, by inoculating into new medium. So callus can be grown easily. So this is one part of it. Only thing is you take a part of it and you do a inoculation onto a fresh medium, that will, so that we can keep this growth of callus, that growth can be easily maintained. All right. Now from this, it is usually, it looks like a mass. Now from this callus we can also isolate individual cells. So how do we do from the callus we can get an individual cell? Now by transferring that into a liquid medium, and we are subject this particular callus into some amount of an agitation. So from here, so we also treat this particular callus also for enzymes. So from the callus, in order to get these cells, we have to use the enzyme pectinase. Pectinase is the one which will digest the the other material which holds these plant cells, in fact, like extracellular matrix, what we call in case of an animal cells. We also have a lot of extracellular components holding these plant cells. So in order to make these cells loose, so we are going to use this one, the enzyme pectinase. So we are going to get the individual cells. So now these individual cells are grown in a liquid medium, and again with subsequent inoculations to a liquid medium, so we can maintain these particular cells in the laboratory conditions. So callus can be maintained easily, and cells also can be maintained easily by providing or by inoculating them into a new, either a solid medium or a liquid medium. Now these cells, we are going to convert them into a protoplast. So what the easiest one is that you take these cells, treat them with the enzyme cellulase, so we are going to get the protoplast. So from an explant, we have worked out how we are going to get a protoplast. All right. Now we are going to subject this protoplast for all kind of manipulations, either a somatic cell fusion, or in case of a recombinant DNA technology, a gene is going to be introduced using a vector. All right. So once we manipulated this kind of a protoplast, now how do we convert this protoplast into a plantlets? All right, in a laboratory conditions. So the manipulated protoplast, again we inoculate them onto a solid nutrient medium. So now this particular protoplast, so because of the nutrient mediums, will try to develop the cell wall, and so now this particular cell, so now we can we can transfer them into a liquid medium, and with agitation, now the protoplast will become a plant cell. Okay, now this plant cell can be further maintained in a liquid medium by inoculating into the fresh medium. Suppose you take these particular cells, transfer them into a solid medium. So now then these cells will become callus. So here it is an individual cells. So now here these cells will grow as a mass. So probably in order to have a mass, so these things has to secrete some extracellular matrix like pectin and others. So we are going to have a callus. So in order to maintain this particular callus, we have to provide low auxin and low cytokinin plant hormones. So now from this particular callus, so which is again derived from a manipulated protoplast, so we can get a plantlets. So if you want to get a plantlets, you take a callus. Now by altering the concentration of these plant hormones, you can get a plantlet. So now here, under low auxin concentration and increased cytokinin, so what happens is that this plant cell, which is in a callus, usually is transformed into shoots. Shoots are going to be developed under high concentration of cytokinin and low concentration of auxin. So now here the callus has shoots in that. On the other hand, you take the same callus cells, then if you under increased concentration of auxin and low concentration of cytokinin, so now this particular callus do develop roots. So now you understand that for the development of roots, auxin is important. For the development of shoot, cytokinase is important. Now under elevated concentration of cytokinin, so we got the shoots. So for these particular shoots, if you increase the concentration of auxin, the shoots do develop roots and convert them into a plantlets. Or here, in case of a callus which develop roots, and if you provide increased concentration of cytokinin, so now the roots do also develop shoots, and we are going to get a plantlets. So from these plantlets, then you slowly transfer them into the land. So now they grow a viable plant. So now these are the various steps. We should know what is a protoplast and how do we get a protoplast. From that, I think we also discussed about like how callus is going to be generated and then individual cells in fact. So we know from an explant, so we can get a protoplast, we can subject this for manipulation, then the manipulation, the protoplast can be converted into an individual plantlets. Alright. So now we will just talk about like what exactly we are going to do in case of a somatic cell fusion. So we take a somatic cell, probably from an explant region. So we make the protoplast. Now once we are going to make the protoplast, probably we are going to take two different protoplasts from different variety. So assuming that one is a high yielding variety, another one is a disease resistant variety. So we have the protoplast from these cells. So now you take these cells. Now in the presence of polyethylene glycan, so there will be a fusion of cells takes place. The chromosomes are going to be combined in this case. So I think we do have chromosomes having the trait of a high yield variety and also a trait of a resistant variety. So we are combining both these chromosomes and then we are trying to get a viable plantlets which is both high yield and resistant variety. Alright. Suppose like in the genetics you have discussed or you have learned the terminology what is known as an autopolyploidy. Another one is going to be an allopolyploidy. So in case of an autopolyploidy, what happens is that usually we have a haploid and a diploid. So in case of a diploid, usually we have a 2n chromosomal number. Now this 2n in plants, not in animals, it is possible so that we can increase this chromosomal number from 2n to 4n, 4n to 8n. These things are possible in case of a plant. So if you do take the 2n and if you increase this to a 4n, so you are going to have the same characteristic feature. However, suppose if there is a seed or a flower of a particular size, in case of a 4n, so the size is going to be increased. So what we call this as a hybrid variety. Alright. So now if you do have a somatic cell fusion from the same species, where 2n is increased to 4n, 4n to 8n, we call this as an autopolyploidy. On the other hand, you take the cells from two different species but more or less related. Now then when you create a plantlet by fusing two different species, we call this as an allopolyploidy. See, both autopolyploidy and allopolyploidy are common in nature. So if you try to screen most of the genetic material in a forest, so you will also find out a lineage and there is new species are going to be created, and the new species is created, we all know it is basically because of the allopolyploidy. It is a natural phenomena, but it is a slow phenomena. But now what happens, people have understood how autopolyploidy and allopolyploidy is generated. Now how that can be created in a laboratory conditions. So as a result, so now both autopolyploidy as well as allopolyploidies are possible to do that in the system. So as a result, because of the somatic cell fusion, several hybrid variety of plants are developed. So we will discuss this auto as well as allopolyploidy in the coming videos.